The baseline requirement for human sexual reproduction is the creation and eventual fusion of two haploid gametes. Through meiosis, diploid cells have their genetic material to form these highly specialized reproductive cells. The anatomical structures required for this process are present from birth, but they remain dormant throughout childhood. The endocrine system initiates the activation sequence. During puberty, testosterone in males and estrogen in females enter the bloodstream, driving the physical changes associated with secondary sexual characteristics. Internally, those same chemical signals trigger the gonads to begin active gametogenesis, initiating spermatogenesis in males and oogenesis in females. Human reproduction is a time-sensitive, systems-level process governed by precise hormonal feedback loops. Understanding the system requires viewing the anatomical organs as structures that function only when activated and regulated by specific endocrine signals. The male reproductive system operates on a logic of continuous, high-volume production. It is a transport line optimized to manufacture and deliver millions of sperm. This diagram of the male reproductive system shows the primary gonads, the testes, held in the scrotum. This external positioning maintains the testes at a temperature 2 degrees below normal body heat, which is the specific thermal requirement for optimal sperm production. Within the testes, the presence of testosterone drives spermatogenesis. The organ serves a dual purpose, both secreting the hormone and building the gamete. Once formed, the immature cells move into the epididymis. They are stored here while they mature. During ejaculation, the sperm travel up the vas deferens, or sperm duct. This is the primary transport route leading away from the epididymis and back into the pelvic cavity. Along this route, a series of glands add vital fluids to create semen. First, the seminal vesicle secretes a nutrient-rich fluid containing fructose, providing the energy required for the sperm cells to swim. Next, the prostate gland contributes an alkaline fluid to neutralize the acidic conditions of the vagina, which would otherwise kill the sperm. Finally, Cowper's gland produces mucus to aid movement. The semen then exits through the urethra. In males, this structure acts as a common exit tube for both semen and urine. The final gamete is a highly specialized vehicle. At the tip, the acrosome contains enzymes to digest the outer wall of the egg cell. Behind that is the haploid nucleus, followed by a middle section dense with mitochondria to power the tail for swimming. The male anatomical pathway functions as a sequential, temperature-controlled assembly line, preparing microscopic delivery vehicles for a hazardous journey. The female system operates under a distinct physiological logic. It is optimized for the cyclical production of a single ovum and the long-term structural support of a fetus. Looking at the female reproductive system, the ovaries serve as the production sites. The fallopian tubes connect the ovaries to the uterus, acting as the transit route and the site of fertilization. The uterus, lined by the endometrium, is the gestation chamber, while the cervix and vagina function as the reception passage and birth canal. These structures are governed by a 28-day cycle. This chart maps the fluctuating hormone levels that synchronize the ovarian cycle and the uterine cycle. Between days 1 and 7, hormone levels are low. The inner lining of the uterus from the previous cycle breaks down and is released as menstruation. Simultaneously in the ovaries, new primary follicles begin developing. Moving into days 8 through 13, the pituitary gland increases secretion of follicle-stimulating hormone, or FSH. This signal stimulates a primary follicle to mature into a Graafian follicle. This Graafian follicle houses the maturing egg cell and simultaneously secretes estrogen into the bloodstream. That rising estrogen travels to the uterus, stimulating the endometrium to become thicker and more vascular in preparation for potential pregnancy. On day 14, a sharp spike in luteinizing hormone, or LH, triggers the mature Graafian follicle to burst. This event is ovulation. The released ovum is swept into the fallopian tube, which serves as the exclusive site for fertilization. From days 15 to 28, the remnants of the ruptured Graafian follicle transform into a new structure called the corpus luteum. The corpus luteum secretes progesterone, which initiates negative feedback. This inhibits further FSH production, ensuring no new follicles develop while an egg is in transit. Simultaneously, progesterone targets the uterus, hypervascularizing the endometrium to its maximum thickness to receive an embryo. The menstrual cycle is a timed biological countdown, ensuring a uterine environment is prepared at the exact moment of gamete release while preventing a second ovulation. At day 28, the cycle reaches a timeline fork. If fertilization does not occur, the corpus luteum shrinks, progesterone levels drop, the endometrium breaks down, and the cycle resets. Alternatively, if copulation occurs, sperm cells navigate the vagina and cervix to reach the fallopian tube. Upon contact, the acrosome releases its enzymes to digest the outer wall of the egg, allowing the haploid nuclei of the two cells to fuse. This fusion creates a diploid zygote, which begins dividing through mitosis into a cluster called a morula as it travels toward the uterus. Once it reaches the uterus, the embryo embeds itself into the thick endometrium. This implantation sends signals that prevent the corpus luteum from degenerating. Because the corpus luteum remains active, progesterone levels stay elevated. This halts the menstrual cycle and maintains the uterine lining to sustain the pregnancy. As gestation begins, the outer layer of the embryo forms the chorion, developing villi that grow into the uterine wall to establish the embryonic side of the placenta. An internal membrane called the amnion surrounds the fetus, filling with amniotic fluid. This creates a buoyant environment that acts as a shock absorber. The mature placenta and umbilical cord become the vascular interface for exchanging nutrients, oxygen, and waste without ever mixing the maternal and fetal blood supplies. Human reproduction is a cascade where precise anatomical structures are activated, regulated by rhythmic endocrine cycles, and ultimately repurposed by a new genetic entity to sustain life.